Yeah, very nice talks. Thank you for the speakers. And so now let's move to the question session. So I have two questions from the audience. So the first question is for the first speaker, Kangjin. And
Yes.
the question is that how much variability in the exposure did the instrumental variable explain when you do Mendelian randomization study?
Okay, so I think the questioner is talking about the MR assumption with instrument.
Mm-hmm.
So the one thing we checked about weak instrument assumption is the genetic variance p-values were less than 10 to the minus 10 to the power of minus 6. And that's one thing. And we are working on supporting that, support that they are not weak instruments, including calculating F values and R squares too. Right now, we don't have idea of F value or R square right now.
So in your Mendelian randomization, if as an instrument of the genus, have you? So how many number of variants did you pick?
That's 5.
5. Okay.
Yeah. Eliminating LD structure. LD related p values. Yeah.
I see.
Okay.
All right. So the next question is for Dongjing. The question is that in your T1 study, did you use the subject level genotype or summary statistics from GWAS? And also, what tool did you use?
Thanks for the question. So we used the gene expression prediction model trained by PredictScan or Metascan, and we used the individual level genotypes to impute gene expression and did our own association test with the facial morphology. Well, we'd love to use the summary statistics, but one thing that prevents me from doing so is because the facial morphology is a multivariate phenotype. And as far as I explored the tool, seems like multivariate association test is not an option. So we have to go with the individual level genotype data.
And also a follow-up question from me is that what tissue did you use?
Yeah, that's a very important question. So a human face is really a highly multi-partite structure consisting of several tissues and cell types, including cartilage, bone, muscle, fat tissue, connective tissue. And we select for 4 GTEx tissues that are highly relevant to face, including muscle skeletal tissue, adipose tissue, fibroblasts, and also brain cortex. So the muscle skeletal tissue represents the skull, the bone, and facial muscles, and adipose and fibroblasts are the principal active tissues that forms the connective and soft tissue of that forms the base of the skull and face. Brain cortex is because it has an influence on the shape of forehead. So we choose these 4 tissues.
I do have one more question for you from the audience. So the question is that, did you account for the mixture markers to control for bias for race-specific markers?
We used the genetic ancestry PC, the principal components, to account for possible population stratification. And this step was specifically done in the phenotyping approach, which is we correct the multivariate phenotype using these genetic PCs.
Thank you.
Thanks.
I have 2 more questions for Dr. Bu. So from the audience, in the CPL, affected CPL design, did you assume that IBD is higher in the affected individuals? And also, have you compared affected and unaffected siblings?
Yeah, we assume that we can estimate IBD sharing in the affected siblings using exome or whole genome sequencing data. And we assume that within the region where we're looking at the rare variants, there's no recombination, so that the IBD sharing applies to the rare variant region. So if there's no rare variant association, then the IBD should be as expected under inheritance. But if there is rare variant association in the region, what we see is that the IBD distribution shifts towards higher sharing. So instead of being like 25, 50, 25% in the region, it shifts towards 20, 50, 30%, at least in our simulations. The second question about affected and unaffected. We were assuming a design in which only the affected siblings are sequenced. So we don't have any information on unaffected siblings. So the short answer to the question is no, we didn't compare affected and unaffected siblings.
So, any other questions from the panelists?
I have one question for Kangjin Kim. There's quite an interesting result, but it would be obviously very nice to be able to validate that.
Mm-hmm.
Is that something that you have plans to do?
I think you're talking about replication study. Yeah, that's what we did. We'll try to do, but so I applied. I mean, I used another 2 different data sets related to type 2 diabetes, but I failed to find that specific genus in there because if you know about metagenomic studies, it is really hard to replicate the result. In the case of the target genus is really rare. So like I said, the genus was uncultured, so replication study was too hard for me. That's why I conducted MR analysis and other network analysis. So right now it is hard for the replication study. Mm-hmm.
I have a question for Dongjing. It's a very interesting talk, Dongjing.
Thanks.
So I had a question about your phenotypes. So you said it's a multivariate phenotype, the facial morphology.
Mm-hmm.
How many phenotypes you really had, and how correlated they are? Can you talk a little bit about that?
That's a really good question. So, well, as you know, the facial morphology is an imaging phenotype. So what we have is the face surface image of each subject. And the phenotyping approach we use is called global to local facial segmentation, which is that we partition the full face into smaller segments. And we have a total of 63 facial segments. When we try to calculate the number of independent modules, independent segments, we get a number around 40. So which means that there is a certain level of correlation among these segments, which are also understandable because they are all facial areas and they should share some embryonic origins. So does that answer the question?
Sorry, I'm muted. Sure. So you had about 40 phenotypes and you analyzed them separately. But
Exactly.
there's facial correlation, I'm guessing.
Mm-hmm.
Yeah, that's one more question from audience for Dongjing. So what's the heritability for the facial morphology?
Oh, that's a really good question. So, well, we know face is is under high genetic control just by looking like our parents or siblings. But it's the heritability of facial traits also vary according to where you look at. For example, the nose area is among one of the facial region that has a really high heritability can be as high as 80%, whereas some other like facial regions may have a heritability as low as 20 to 30%, but I think a range from like 25 to 80%, it's a typical range for facial morphology.
Thank you. Yeah, that's very informative. Yeah, audience have one more question for Shirley. And so for rare variant, the rare variant sharing transmission, when comparing a rare variant versus common variant, does the rate of transmission are different? And how does that impact the analysis?
I don't know if I can answer that question. I mean, the transmission of the polygenic risk score is going to be kind of some average over whatever part of the genome you're actually capturing the common variants from. So it's in a sense, it should maybe be more diluted than the rare variant transmission is, but yeah, I can't answer that question more precisely.
So any other questions from the panelists? Or if anyone has questions related to all the 8 talks, yeah, please bring it up.
Perhaps in that case, we'll say a very warm thank you to all of the speakers in the session. And
it was a great job. Yeah.
Very nicely done. And we will hopefully see you all for the next session, which is tomorrow for me, with the keynote speakers and the Williams finalists coming up soon.